Prayer is to the morning, and prayer is the key of the morning and bolt of the evening. So, life is a beautiful movie. Life is a beautiful movie where you write your ending. So, keep pretending, keep writing. Very well said by Jim Henson. Good evening, ladies and gentlemen, for this last day of our FDP. Our 1 week online FDP, which started on 1st of November 2020, is on its landmark on ending. And today is a special day as well because we have learned a lot of things from this online FDP. And with this learning experience, I would be thankful to all the organizers, especially IJMS, who collaborated with Sushila Shankarrao, Gadhave Mahavidyalaya, District Khandala, District Satara. And today we are having a guest speaker for the day, and he is Mr. Mahesh Balwant Khot, postdoctoral researcher, principal investigator, Fondasite, the National Fund for Scientific and Technological Development, Government of Child, host Biotechnology Center, University of Concepcion, Chile. I welcome you on board, sir. It gives me immense pleasure to welcome you for this FDP of ours so that you can share your thoughts and your experiences on our topic of the FDP, statistical analysis in academics and research. And now I present you Dr. Mahesh Balwant Khot, and over to you, sir.
Yeah. Thank you, Dr. Alia, uh, for the introduction, and also thank Dr. Ashok and the team behind International Journal of Microbial Sciences for inviting me today for the motivational talk. And I hope I will be able to motivate the members, the attendees through my talk. I will be sharing my presentation soon. I hope it's visible.
Yes, sir, it's visible.
Yeah. So uh, as a topic of this faculty development program is about statistical analysis in academics and research, I'm going to share my own personal experience with statistics uh, in my last 10 to 15 years of professional career. I had been in hardcore research in microbiology since 15 years. Before PhD and even after PhD, I'm getting encountered with different areas of statistics. Being from the microbiology background, I mostly dealt with biostatistics, but the principles remain the same whether you change from chemistry, physics, or other sciences. So I'm going to share my interaction with statistics and how it is an integral part of an academics. The whenever we hear the term statistics, it's kind of a certain kind of fear we have in mind. It's complicated branch of mathematics, as you are aware. And the most important thing is it is hard to learn. It's not easy. There are reasons why most of the people consider statistics as hard to learn or to understand. So I have noted down some points over here. First point is statistics is different from the probability. Although statistics, basically the results which we obtain in statistics are with the help of probability theory, the logic of probability is totally opposite to the logic of statistics. In stat, we go from sample to population. We have a limited data set, and with that data set, we make general conclusions about the population. But in probability, we go opposite. We have a general view of the scenario, and using probability theories, we compute the various outcomes. So it goes from population to sample. Uh, Other aspect of statistics is every field, every field has some technological terms. So even stat has some technical terms which has specific meanings. Now, in stat, we normally use terms like significance, error, confidence, or hypothesis. In day-to-day life, we are familiar with these terms, but in statistics, these terms carry specific meanings. The meaning which we use in day-to-day life is totally different when it comes to statistics. So it is necessary to learn the statistical meaning of these terms. The problem is that the problem is not that we don't understand these terms. The problem is that we assume that we know what do you mean by hypothesis, what do you mean by confidence, and we go with the misunderstood meaning of these terms. So it is necessary to understand the statistical basis. When we say the results are statistically significant, we should know the statistical basis of that. And the third point is, as it is a branch of mathematics, many people think that you should be proficient in mathematics to understand statistics. But frankly speaking, when it comes to applying statistics in research, you don't need to have a deep knowledge of mathematics. All you need, you should have a conscience on which methods you should follow in analyzing your data. If you have a sound knowledge of that, you can effectively use statistics. So basically, you should be able to use statistics in an effective way for your data set without deep knowledge of mathematics. Uh, Now, as far as lab research is concerned, mostly we deal with whether it's physics, chemistry, or biology, or engineering field as well. We do plan our experiments, and the scientific approach to experiment planning is very important. And now I'm going to tell you where statistics fits in this scenario. So these are the guidelines basically which I have noted down from my own experience. These are around 6 to 7 recommended guidelines on how to use statistics in your experiment designing and analysis. The first and most important step is you need to recognize what is your problem. Once you define the problem for which experimentation is required, you can make a clear statement. Now, you may think that it is an obvious point, but most of the times we may not realize what is the actual problem which you're going to study. So, once you define the problem, you can bring the solutions to the problem in a proper way. Once you define the problem, the next step is to identify the factors which are known to influence the performance of the system or the problem which are going to study. Once you identify the factors, the next step is the level on which these factors you're going to vary, the range. And once you define the range, you have to choose the levels. Normally we use 2 levels. Minimum levels should be used for each factor, low level and high level. Now, the third thing is to select the response variable. This is the variable which is mostly the mean or standard deviation of the response. So these 3 steps, the first 3 steps, recognizing the problem, selecting the factors, and response variable, are known as pre-experimental planning. Before the actual experiment begins, these 3 steps are important. Once you define these 3 steps, it's easy to go ahead. The next step is obviously you have to use the experimental design. Now here, the role of sophisticated statistical software comes. We have a lot of software packages which help us to choose experimental design. Now, this is also a critical step. If you have done the experimental planning properly, it is easy to go for the proper experimental design. And statistical softwares help us to choose experimental design. Normally in software package, you need to enter the factors, the levels, the ranges, and then we get the worksheet. Like in Excel, we have a worksheet. These software packages also gives us a worksheet, and you can enter. And you don't have to do anything. The software gives you the two options. Either it will give the recommended design, or it will give the selection of different experimental designs, and you have to choose from one of them. So if you have done your pre-experimental planning, if you have selected your factors wisely, if you have selected, if you identified your problem carefully, the experimental design selection is not a big deal. It is a very straightforward thing. The next step is obviously you have to perform the experiment as per the design. You have to make sure you are performing the experiment as per the design. Then once that is experiment has been performed, you have to use different methods of statistics to analyze the data obtained in the experiment. Now here also again the role of statistical software comes. They gives us an interface to interact with data and to analyze the data with different available statistical tools. And at the end of statistical analysis, the main aim of statistical analysis is to obtain result and conclusions. That is the last step. The result and conclusions should not be judgmental, but should be objective. So basically, that gives an idea how much uncertainty is there, how much error is there in your experimentation. So if you use statistical approach in your experimental designing and data analysis, your whole research becomes valid. You can draw concrete objective conclusions if your experiment is prone to errors. So statistics helps a lot in designing experiment followed by data analysis when you're researching something. This I'm going to explain with my own work, how I used statistics in my own research. This is the paper I have published way back in during my PhD in 2015. Here, the objective or the definition of my problem was optimize or to increase the production of FAME or fatty acid methyl esters from a biomass of oleaginous fungus. So that was the problem I defined. Now, after defining the problem, I identified the factors which are known. I had a knowledge of which factors are known to affect the biomass production. So I've enlisted factors, as you can see in the table, temperature, time. As the process involves catalyst and some chemicals, I have added catalyst, then I have added reactants, that is methanol, chloroform, biomass, and other physical and chemical factors I have added. You are also able to see, I have used two levels, -1 and +1. These are nothing but low level and high levels. At two levels, I have taken all these factors. And if you see, for example, temperature, I have taken a range. That range is 50 to 80. Then for catalyst, I have taken 1 to 10 ml. So basically, that is the range you should know. You should have some previous knowledge about the problem you're going to study. So you have to select a range and you have to select the two levels. And this design, these factors, then I have put into the this data have been fed into the statistical software. Many of you might have used this software which I've mentioned in this paper, that is Design Expert software released by Statis. And here you have to enter the factors levels, you have to give information to the software, and then it gives us the design. In the table 3, you can see the design matrix basically, and the design matrix gives us the experimental runs to be carried out. And the response variable, as I've said, my problem was about to optimize the production of fatty acid methyl ester, so that the production quantity was the response variable, which is at the last column of the table 3. That was my response variable. And the factors are varied at different permutations, combinations, and the design matrix is generated, which is giving me the 12 runs. Now, experiment was performed according to the design matrix, and the data was analyzed. ANOVA was used to, as per the software analysis, and data analysis has been done. So basically, what you have to bear in mind for the experiment, that these are the 3 to 4 points which are very important to keep in mind, that you have to think beyond statistics when you are doing some research. As an experimenter, as a researcher, you are expert in your field. So you have to use your conscience, and you have to use all the possible outcomes, all the possible, you should know the objectives of the research you're going to do. That most of the time it happens that you get overboard, you get overzealous, overexcited about the statistical designs, and you might tend to use complicated designs, because software gives hundreds of designs, but it's up to you to use the design which is as simple as possible. So because that avoids the problems further. That is, you might encounter problem in data analysis. If your design is simple, data analysis is also straightforward. Then the most important problem is that most of researchers don't understand the actual meaning of significance. So a significance in statistics carries very different meaning. So we should know the difference between statistical significance and practical significance. For example, 2 means might be statistically different, but doesn't mean that it will have a biological or chemical relevance. So while saying the result is significant, you have to be very careful, because it might give a wrong message. So you have to know the difference between experimental significance and statistical significance. So these are some points which experiment need to take in mind. So as I talked about the statistical significance, I would like to give one example of how the statistical significance is important in research. Like this paper was my first paper during PhD, and I had a problem when the the reviewers sent back my paper asking what is the statistical significance of your particular data analysis. And they wanted to know, as I've shown in figure 2, they wanted to know how the means differ significantly from each other. So at that time, I learned how to use hypothesis testing in your research. And I use one-way ANOVA followed by some multiple comparison test. And I was able to compute how the 2 samples differ from each other statistically and biologically as well. For example, if you go in detail about this figure, I have taken 5 different fungal strains, and I wanted to see how much total lipid produced by these 5 fungal strains differ with each other. Uh, if you see the superscripts on the top of the dark green colored columns, some share different superscript letters like A, B, or one column is showing A, B. So this A, B means it is sharing the mean with other columns representing mean by A and some columns represented by B. So to compute this, to know this, ANOVA helped me, and ANOVA followed by multiple comparison tests, that is Tukey, which Tukey multiple comparison test, which most of us have used, helped me to compute this important observation, and it helped me. This is another paper where I used multiple comparison test to know the difference between different extraction methods which are used to extract lipids. As my research area was on single cell oil, that is a total lipid from microbial cells, my aim was to test different methods as mentioned in this figure 2A, B, C, D, E, and I wanted to check which of these methods have influence on the yield. So I did ANOVA followed by multiple comparison tests. Each mean was compared with each other, and statistical significance was calculated. And as you will see, most of these are different from each other, but the red colored columns like B and E are not statistically different, so because they carry the same mean, that is varying within the same range. So this is how the hypothesis testing helps us to draw conclusions, and biological significance, its relevance to biological significance can be computed. As I mentioned about statistical softwares, my personal experience I'm going to share. I'm using mostly the non-sophisticated softwares, like which are easy to use, user-friendly softwares like Prism. I'm using this software since almost 5-6 years, and I'm not endorsing Prism here, but I'm sharing my own personal experience that it is easy to use, user-friendly. I have shared screenshot from the actual software analysis where you can see the actual how you enter the data and what types of test you can do with this software. Like you can do t-test if you have two means, paired, unpaired, it gives you option. If you have more than two means, you can obviously go for ANOVA. Then you also have other statistical analysis tools available, like if someone is studying enzyme kinetics, if you want to study Michaelis-Menten or competitive inhibition studies, even kinetic studies can be done with this software package. And they have special academic packages, group subscriptions are also available. And the point of telling of this is I'm using this software package, and it helps a lot. It also helps you to graph your data, to graphically present your data, and publishable results you can generate, and it is user friendly. But apart from this, earlier there are also many software packages available in the market. I have used earlier SPSS, which is very professional software developed by IBM. The only problem with spaces, it is quite expensive, but if you have enough funds to cover this, you can definitely go for spaces. My personal issue with spaces, it is not that user friendly as Prism by GraphPad is. Spaces is like you need to get hands on this software. Once you are familiar with this software can make wonders, because it is used by worldwide in almost all fields beyond science. It has been used. Then there are also old players like Minitab, MATLAB, which are mostly used by mathematicians. So these are the also softwares. I haven't used MATLAB or Minitab. My own experiences with SPSS and Prism. So yeah, so that was my personal experience with statistics. I used statistics in designing, and in designing the experiment, followed by analysis of the data, and it helped me. And if you have take home story is, if you have a good software with you, and if you know which methods to use, statistics definitely helps in your research, and you become more proficient, you become more expert in your work. So that's all. Thank you, International Journal of Microbial Sciences, for inviting me for this motivational talk. And if you have any questions, please feel free to ask. Thank you.
Yes, sir. Thank you so much for your insights on the topic, and I hope all our participants would have definitely benefited from this. And now I again tell that statistics is a part of research, and research without statistics is like we are not having the salt in our food.
Yeah.
So exactly that's what I understood, because I am a person from non-statistics background. So this is like an eye opener, and I think you have summed up all the things whatever we have been learning through all these 7 days. So it was really good. I thank you personally as well as from the organizers side also to make your time and give your presence for this valuable FDP of ours. Thank you once again, sir. Now
Thank
move.
you for having.
Yeah. Now moving on towards our technical session of the day. Before we move on to our technical session, I would like to make some notifications for the participants here that our FDP is for the 7 days, and today is the seventh day. That means we have traveled through these 7 days, learning the statistical analysis in research and academics. And this is crucial for everybody as a researcher, as a PhD scholar, or in academics to learn and to imbibe. So, with this thought, I think we have one more thing for you all, that is MCQ test of 50 marks, which is mandatory to all the participants to receive their certifications. Out of that 50 marks, they have to score 20 marks, which will be like their eligibility criteria for getting the certifications. All the links are on the website, that is www.theijmspublic.com, the official website of ours. I repeat, www.theijms.com, to visit this website for regular updates and links. Feedback link and the MCQ link is already posted. It will be opened or able tomorrow, that is 8th November 2020, sharp 7 p.m. So I request all the participants to utilize this opportunity. We are going to open 3 links at the same time so that all the participants can be benefited. So there will be no kind of we have a limited number, so we cannot do it. We will not have that issue. So this will be a very good plus point here. And the next best thing is the organizers have decided the best participants, the best 3 participants will be getting the national level award. So I hope you will all be interested in getting an award for yourself, which will be delivered to your postal address. And the best speakers award is also there, and that is national level award also for the best speakers for the presentations who have given their extreme time and their kind of expertise in the subject knowledge sharing. So, with this, I would be moving ahead towards our first technical session for the day 7. And the topic for today is sample size calculation in research. And the speaker for the day is Professor Ramkrishna Lahu Shinde sir, Department of Statistics, KBC North Maharashtra University, Jalgaon. And I welcome you, sir, on board. Before we proceed, I would like Minal Majati to give the speaker's profile to all our participants to know better. So over to you, Minal.
Good evening, madam. Shall I start?
Sir, there is an introduction of speakers.
Oh, I see.
So yeah, we would be wanting to do that. So Minal, are you online? Can you hear me? Minal, I think we have some connectivity issue here, sir. Minal, sir, I think we are facing some connectivity issue here. Yes, sir. You can carry on, sir. We lost her, I guess.
Okay. Good evening to all of you. Let me share my screen.
Yes, sir.
Uh, can you see my screen,
Yes, sir.
madam?
Your screen is visible, sir,
Yes.
and you're audible
Uh,
as well.
First of all, I would like to thanks to organizers for inviting me to deliver this lecture. In fact, I have delivered many lectures in the research methodology course of our university. Around 1,000 students attended, and many lectures related with research methods, research objectives, then statistics, sampling theory, and this is one of the lecture out of those lectures related with sample size calculation. So as I know that most of you are from the life science background, participants are from the life science background. So I will try to make it simple to calculate the sample size. How does the sample size relates with the standard deviation and variance, that we'll like to see. Myself, Professor R.L. Shinde, Head Department of Statistics of Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon. I have been working here from last 27 years. Uh, and I will start my presentation related with the topic because we have limited time. Estimation of population mean and population proportion, confidence interval, and sample size. In fact, the lecture is for calculating sample size, minimum required sample size we want to deliver. But before we go to sample size, we should have some background of estimation of population mean and population proportion. Why? Most of the time, our interest lies in estimating mean and estimating proportion. In almost all fields, social sciences or scientific pure sciences, in many fields our interest lies in comparing population means or population proportions. Statistics itself a science of comparison. So uh, in order to compare those, we need a minimum required sample size. So how does the sample size is calculated? That is our third part. Before that, we will try to talk about estimation of population mean and population proportion under simple random sampling. So when we have a finite population, suppose we have a finite population of size capital N, and we want to draw the sample, simple random sample of size small n without replacement. That means those units are drawn one by one. Units will draw. Suppose we have capital N sheets, they are numbered from 1 to capital N, and one by one we will draw till small n sheets are drawn, and those drawn elements are considered as the sample elements, and their y values, the suppose there are capital N students and we want to select small n students, so we'll put those sheets there and we'll draw n sheets, and their roll numbers will be written over there. And what is y variable? Y variable is suppose their marks obtained or their height or their weight, whatever it may be. So y variable, suppose their student's height. So small y1 is nothing but the height of the first student in that population. Small y2, height of the second student. In this way, sum of all yi's divided by capital N, that is the population mean. This is our population mean. This is unknown to us. This is unknown to us. Suppose there are capital N people in the state, and their average height I want to estimate. Their average height is unknown to me. All those people in the Maharashtra state or India, their average height is unknown to me. So because all people's height is not known to me. So y bar, small y bar, that is the population mean. And our objective is to estimate this population mean. So the unbiased estimator in the statistical theory is sample mean for this population mean. You draw only n elements from the population and consider their y values. Y value means response variable or study variables value. So n elements average y value, that is sample mean, and this we treat as a estimator for the population mean. That is it is unbiased estimator. That means expected value on an average value of capital Y bar is small y bar. We'll demonstrate through example also this. Note that the sample mean changes as the sample changes. As sample changes, sample mean also changes. This is very important feature of statistics, because suppose you consider example of, I am taking here purposefully small example, small capital N is equal to 6. There are 6 villages suppose, and we have to take the sample of 2 only. We are going to draw only sample of 2. How many possible samples of 2 villages out of 6 villages are possible? That is 6 choose 2 or 6 C2 in mathematics we call. That is 15 possible samples. I have written on the next slide all those 15 possible samples. 1, 2, 1, 3, 1, 4, 1, 5, 1, 6. Then 2, 3, 2, 4, 2, 5, like that. So all 15 possible samples one can write down. Under simple random sampling, we can have out of 6, if you choose 2, there are 15 possible samples. I hope you understand this. Study variable y is here number of cows available in the village. A simple example we have taken. Number of cows. Our interest lies in estimating average number of cows available in a village. There are 6 villages. We want to estimate population mean, small y bar, average number of cows available in a village. That means total of all villages cows divided by 6. That is unknown to us suppose, and that we want to estimate. Now for a dummy study, we assume that all 6 villages number of cows are available with us. Suppose these numbers are available. For example, the first village has got 400 cows, second village has got 300 cows, third 200, fourth 1000, fifth village 800 cows, and sixth village 600 cows. In fact, we know the population mean here. Population mean comes out to be 550. Sum of all these divided by 6 very easily. Population total, how many cows are there in all 6 villages? That is 3300. This is population total. So what are we looking for? We are looking for, if we take a sample of 2 only to estimate this population mean, to estimate the population mean. In fact, this is for dummy study, and where we know the population mean also. But suppose sample of 2 only taken, So what are the all possible samples? Sample population element 1 and 2. That means village number 1 and village number 2 are selected in a sample. Then the sample mean is 400 plus 300 divided by 2. That is my sample mean 350. Like that, if sample is 1, 3, so y1 plus y3 divided by 2, that is 300. That is sample mean. As sample changes, the sample mean is changing. So minimum sample mean obtained for the sample 2, 3. If the village number 2 and village number 3 got selected in my sample of size 2, the sample mean is 250. And estimated total population can also be obtained. How do you obtain total population? Sample mean multiplied by capital N. That is, on an average, 350 cows are there. If this will be your estimate, if sample is selected 1, 2. 1, 2 is the sample selected. Then we have estimated population mean by 350. In fact, true population mean is 550, but this is the sample taken by you. Then it is showing you the population mean estimated is 350 multiplied by capital N, that is 6. So there are 2100 cows in these 6 villages. This is our estimate. As a statistician, we have given this much because we are not known about these 6. All YIs are unknown. For some time, we'll assume that all YIs are not known to you. Only these 2 YIs are known. So in this manner, if all samples, there are 15 possible samples: 1, 2, 1, 3, 1, 4, 1, 5, 1, 6, 2, 3, 2, 4, 2, 5, 2, 6, then 3, 4, 3, 5, 3, 6, 4, 5, like that. So 15 possible samples. Note that the population mean, exact population mean, true population mean is 550, and as sample changes, estimate is changing. This is the fundamental theory of statistics and sampling theory also. So sometime we overestimate. For example, there are 550 cows on an average in 6 villages, and if you take sample selected is 4, 5, then 900 cows are there on an average per village. This is your estimate, that is overestimate. If you select this sample 2, 3, then you underestimate. 350 also you underestimate. Exactly 550, there is one sample 2, 5, which gives you sample mean as well as population mean same, but you are unknown because all 6 values may not be known to you before you sample. Okay, only 2 values are known to you. So even though your sample mean is exactly matching with the population mean, you are not known that okay. So as sample changes, sample mean changes. Then variance of the so if you consider all these sample means 350, 300, 700, 600, as sample changes, sample mean changes. There are 15 possible sample means. If you take the average of these all sample means, it is exactly 550. Mean of sample mean is equal to 550. Mean, that is what we say that the sample mean is unbiased estimator of population mean. Mean of all possible samples, sample mean is matching with population mean. That is the criteria of unbiasedness. But it is not the case always we will do all possible samples and write like this. This is only dummy study in order to teach you, okay, for fundamental concept. So if you talk about the variance of all these values of y bar sample mean, that is measured in statistics by this formula 1 minus f sy square by n for finite population size capital N. If you have infinite population, then it is sy square by. What is sy square? It talks about the population variance of study variable. Number of cows in a village, y1, y2, y6 we have minus 550, that is population mean, its square divided by n minus 1. This is a population mean, population variance of the study. Our study variable is number of cows available in a village, that is study variable. Now f is n by n, that is sampling fraction, and sy is square root of this, that is population standard deviation of the study variable. So unbiased estimator of SY square, this is population variance, which is also unknown to us because we don't know all Y values of the study variable. So how can you know the variance of the population elements? There are 6 villages. I don't know how many cows are there in each village and what is the population variance of that study variable. So it can be estimated by your sample. You have taken a sample of small n, so one can estimate like this. Small yi's are y values of the ith element of the population, ith village number of cows, and yr, capital YR, denotes the village selected at the earth draw. We'll observe the how many cows are there in the earth selected village. That is our sample data. Okay, so capital SY square is my unbiased estimator for the population variance SY square study variable, and accordingly I get the standard error of y bar, that is square root of variances. Then we get the confidence interval for the population mean. Only estimating population mean is very easy. Sample mean, capital Y bar, is a point estimate of population mean. But we need confidence interval in order to judge the whether where is my true value of population mean lies. True value of population means. Capital Y bar is not exactly true value of the population mean. That is sample mean for that sample taken by you. So for that sample, what is the confidence interval? Y bar plus minus Z alpha by 2 standard error. Z alpha by 2 is nothing but the point on the normal standard normal distribution. Somebody might have taught you distributions, binomial, normal. So it is a right hand side point. Above this point, remaining probability is alpha by 2 for standard normal distribution with mean 0 and variance 1 or standard deviation 1. That point, that table value can be seen from the table I have given on the next slide. T distribution, when the sample size is small, we have to use confidence interval like this: y bar plus minus t alpha by 2 for n minus 1 degrees of freedom multiplied by standard error, y bar minus this and y bar plus this. Okay, if you want to observe these z values and t values are changing as n changes, and z value is fixed for fixed alpha. Alpha is 0.01, type 1 error. Type 1 error or level of significance is 0.01. Then z alpha by 2 is 2.58, 1.96 for 5% level of significance, and for 10% level of significance it is 1.64. These three values most popularly being used in the statistical theory. You can use these directly, or you can observe through tables. You may find difficulties in observing table, so it is better to note these values for these three level of significance. Generally our level of significance is 1% or 5% many times. So if your sample size is small, in confidence interval on last slide, the instead of z alpha by 2 you have to use t alpha by 2 n minus 1 degrees of freedom. So these points are differing. For example, for n is equal to 2, 5% level of significance, this is 12.7. For n is equal to 31, it is 2.04. As n increases, it is approaching to 1.96. It is approaching to 1.96. So if you have large, some people say that if you have n is greater than 30 or 50, you can use these z values directly. But then also for n is equal to 50 also it is not 1.96. So it is better to use t value for whatever may be sample size. Okay, unless you are given very large sample size. Okay, confidence interval for population mean n is equal to 2. When you use t for 5% level of significance, t value is 12.7. If you, what is the confidence interval? Y bar sample mean minus this t point multiplied by standard error. Similarly, this. So if the sample selected is 1, 2, according to we have already obtained the sample mean in the previous slide. Sample mean plus minus, if you do, these are the confidence intervals. Negative lower confidence limit, you can take this as a 0 also, because average number of cows cannot be negative. Average number of cows in this example cannot be negative. In some examples, if the observations are negative also, study variable observation, then in that case you can keep this as it is. Here in this particular example, average number of cows cannot be negative, so lower confidence limit can be taken as 0. So that if the first sample is selected, so in this interval somewhere the true value of population mean lies. That is what you are concluding. What is the true value of population mean in this particular example? We know the true value of population mean, that is 550. So 550 lies in this interval. 550 lies in second interval also. In all possible samples, in this particular example, 550 is lying in these intervals, confidence intervals, all 15 samples. So that is interesting. So in fact, when we say it is 95% confidence interval, we are supposed to give only confidence that 95% samples will include population mean, true population mean. In fact, here 100% samples are including. This is one small example. In different examples, you will find that some 5% cases you may fail. But here one has to use T point. If you use Z point, that is wrong actually. One should not use Z point. If you use Z point, the confidence interval becomes y bar plus minus Z alpha by 2 standard error. And instead of T point, if you use Z point purposefully, then you are in wrong, your conclusions will be wrong, because for n is equal to 2, T point was 12.7 and Z point is 1.96. So confidence intervals will be differing. And because of this wrongly using Z alpha by 2, 4 samples are such that 1, 2, 1, 3, 2, 3 and 4, 5. Their confidence interval drawn from these 4 samples does not include the population mean. True population mean is 550. This is known in our particular example, because we know in all population elements y values are known to you. Therefore, sample population mean does not lie out of 15 samples in 4 samples. This is not the mistake of statistics, because you are wrongly using statistics. You should not use Z alpha by 2 here. You should have used T, because your sample size is small. So this is I have noted here. What is the meaning of 95% confidence interval? Many people feel that what is the interpretation of 95% confidence interval? If you consider all possible samples, please note, out of, for example, in this example, the population size was 6 and sample size was 2. So all possible samples were 15. But in many situations where the population size is 1000, sample size is 50, then all possible samples will be crores of number, because 1000 C 50 is a big number. So what is the meaning of 95% confidence interval for population mean? It is nothing but, if you consider all possible samples and obtain 95% confidence interval for population mean, as we have obtained in the previous example, 95% samples out of total samples will include the true value of population mean, and only 5% samples will be such that their confidence interval will not include the true population mean. That means what happens in real life, we do not consider all possible samples. In real life, we take only one sample. We never write down all possible samples. This is dummy study. That was the dummy study what we have discussed just now. But in real life, we take only one sample. Suppose there are 1000 students in college. We have taken randomly 50 students. That is a single sample. We never take all possible samples. So in fact, generally our results are based on only one sample. So if your sample is from those 95% possible samples whose confidence interval includes population mean, then you are lucky. What we have said, what is the meaning of confidence interval 95% confidence? If you write down all possible samples of size small n out of capital N, if you write down all possible samples and obtain their confidence interval for population mean, there will be 95% samples whose confidence interval will include population mean, true value of population mean. This dummy study one can conduct for different purposes. And 5% samples will be such that their confidence interval won't include the true value of population mean. So if you take only one sample, and if your sample is coming from those 95% samples, then definitely your confidence interval will include the population mean. If you are lucky enough means you got a proper sample. Proper sample means your sample is belonging to those 95% samples. There are exactly 95% samples in your population whose confidence interval gives you such that it includes the population mean. Note that the probability of getting such proper sample is 0.95. 95% probability is there. So one should not bother about this issue. You are likely to be lucky with probability 0.95. Therefore, we blindly believe on that confidence interval, and we tell the user that in this interval somewhere your population mean lies. Let us consider the example. Suppose there are 10,000 elements in the population and sample size is 400, and survey results show that the sample mean is 55 and sample variance SY square, capital SY square, I have given formula for sample variance, that is 16. Suppose this is the extracted data. Sample mean is 55, sample variance is 16. This is obtained after taking a sample of 400, and now we are asking for the confidence interval. So as the sample size is large enough, here we use Z alpha by 2, y bar plus minus Z alpha by 2 standard error. Okay. So if you use this, Z alpha by 2, alpha is equal to 0.05, that is 95%. Therefore, it is 1.96 Z alpha by 2. Y bar is 55, that is sample mean. And standard error we have obtained here, that is 0.1959. So confidence interval comes out to be 54.61 to 55.38. Such a nice confidence interval. That means very close. Your true value of population mean lies here, and the confidence, if confidence interval is nice, means what? If it has a very less length, smaller length of confidence interval, always better. If you have larger length confidence interval, that means you are saying that your population mean is lying in this interval somewhere. So if this interval length is small, we will be much happy. So for example, if I take in this above example, if you as the population size is very large, you can use the approximated value of standard error also. You can forget 1 minus f quantity. This is closer to 1, because 400 by 10,000, it is closer to 0. So this may you can use 0.2 also instead of 0.19 here. In this case, you can use 0.2. This is just note as capital N was very large population size. Okay. If now suppose in the same example, so instead of 400 sample size, if you have taken only 40 sample size and sample mean is 55, samples variance is 16. Suppose you have obtained the same sample mean and sample variance based on the sample size 40. Are you going to get the same confidence interval? Even though your sample mean and sample standard sample variance are same, your confidence interval is not same, because here confidence interval was 54.6 to 55.38, and now it has become because why it has changed? Because of standard error has changed. Standard error depending on n. your standard error depends on n, because there was sample size 400, now it is 40. Now it is 40 sample size. So standard error has changed here. Instead of 0.19, it is 0.63. So larger standard error, therefore larger length of confidence interval. So we have observed that the sample size decreases, the length of confidence interval decreases. If the sample size decreases, length of confidence interval it should be increases. Sorry, it should be increases. And if sample size increases, the confidence interval decreases. Here it should be increases. I'm sorry, on page number 10 here we should add like this. Okay, then this was the example. Now estimation of population proportion. Many times our interest lies in estimating proportion uh, in many situations, in chemical problems or microbiological problems or social sciences problem. I have taken here simple example like social sciences. For example, proportion of people in favor of politician. He wants to know, politician, what proportion of people are from my side. So what is the unbiased estimator for the P? P is a parameter, proportion of people. Suppose he doesn't know what proportion of people are from his side. He wants to take a sample of size small n, and out of small n, how many are answering that we are from your side or we are going to vote for you. So D is number of people in favor of politician randomly selected by using simple random sampling. And the standard error of this estimator P hat, it is estimated proportion, sample proportion, and this is population proportion P. P is a population parameter, population proportion, which is unknown to us, what proportion of people are from his side, and that is estimated value is D by n, D number of people from his side out of small n. And its formula for finite population 1 minus a p hat q hat upon n minus 1, and for infinite population this 1 minus a becomes almost 1, closer to 1, because f is closer to 0. What is f? N, small n by capital N. If small n is comparatively very small than capital N, it goes to 0. So f becomes 0. So in many situations, your population is infinite. Uh, in some experimental field, population might be finite also. You can use this formula. So what is the confidence interval for P? P hat plus minus Z alpha by 2, standard error of P hat. We have to use these formulas for standard error. Generally, capital N is sufficiently large, uh, and here there is no problem of using T. Generally, while estimating proportions, we use sample size at least 40, 50 or more than that, many times in thousands also. So there is no question of T confusion with T here. We can use Z alpha by 2 directly. For example, suppose the population of 2 lakh people, 200,000 people are there in the population, and you have taken a survey of 1,000 people, and it has been shown that 75% people out of surveyed people are in favor of politician, in favor of particular politician. 75% people are there out of 1,000. Okay, it is estimated, and now you want to give the confidence interval. This is a point estimate. This is a point estimate. You cannot believe on this value directly. You need the confidence. 95% confidence says your population proportion, true value of population proportion, what proportion of people from that politician's point of side, that lies, true value lies somewhere here, 0.72 to 0.77. So he can decide accordingly. At least 72% people are from my side. Okay, I can try this election, something like this. But if this confidence interval is very having large length, something like this, 0.1 to 0.9, that means 10% to 90% people are from my side. So he doesn't know how to take action. Okay, so this confidence interval should be as small as possible. So length should be as small as possible. Okay, another example here, n is equal to 300. Suppose sample size in the same example of capital N is equal to 200,000 people and small n sample size is 300, and suppose the 75% people are from sample 300 from that politician side. So standard error differs now. Standard error quantity was last time 0.013. Now it is 0.025 as the sample size changed. Sample size was 1000 last time and now it is 300. So the confidence interval is 0.70 to 0.79. That means 70% to 79% people are from his side. Somewhere here the true value lies. That is what you are saying to that politician. Suppose now the sample size is only 100 and sample proportion again comes out to be 0.75. I am trying to keep this constant because I want to demonstrate that as the sample size decreases, the length of the confidence interval is increasing. So he may be in dilemma, the person who is getting this information. 66% to 83% vote I am likely to get. Somewhere here my true value lies. But he may be in dilemma in such situation. Suppose these numbers are, for example, 0.16 to 0.23. So he may not take the correct action. He may not be able to take. So small sample size gives you confidence interval of higher length. So one should go for the sufficient sample size. So which sample size is better for us? This is the question again. So that is what we have to tackle such questions. One more example I have given for proportion here. Population size is 2000, sample is 10% of 2000, that is 200 we have taken. Suppose these are the sample sizes were dummy, we have decided, and we are taking working out the confidence interval. Your point estimate is 40%. That means you will tell that around 40% people are likely to give you votes. But he is asking what is the 95% confidence for my true value. So true value lies somewhere here, 33% to 46%. Somewhere in this interval, the proportion of people from your side that lies here. That is what you are saying. Your answer this may be correct if your sample is coming from those 95% correct samples, lucky sample. Now you want to go for the determination of minimum sample size required to desired accuracy under simple random sampling, estimating population. Now this is interesting. This is the point of discussion. Our interest lies in deciding minimum sample size n such that our interest lies in estimating the minimum sample size or deciding minimum sample size n such that the population mean small y bar and sample mean capital. I am using reverse notation. People are generally using capital notation for population. I have used reversely. This is for population mean, capital Y bar, sorry, sample mean, and this is for population. The distance between the population mean and sample mean, absolute distance. For example, this is 25 and this is 26. Then difference is -1, absolute difference is 1. If we want this difference should be less than small d. D is your choice, user's choice, and alpha is also your choice, user choice. So we want to decide a sample size in such a manner that the difference between sample mean and population mean should be less than d with probability at least 1 minus alpha. So in order to solve this problem in statistical language, we have solved this problem in statistics and we come out with the formula that your n should be, sample size should be. This is the user problem. User is saying that the population, you decide, you give me estimate of population mean in such a manner that the difference should be less than d with probability 0.95. Suppose alpha is 0.05 with probability 0.95. So I am ready to give him sample size accordingly. So n should be greater than equal to capital N divided by 1 plus this capital N, we know that is population size. D, this user has given D. What is SY square? That is the population variance of study variable under study. And Z alpha by 2, from alpha, given alpha, we can decide Z alpha by 2. And accordingly, uh, approximation formula is also given, because when capital N is very large, approximation formula for N is like this. Now, SY square is a population variance of study variable. Study variable, for example, in last case, our study variable was number of cows in a village. So there are, suppose, 100 villages. 100 villages cow numbers are put in one column and find its variance. That is the population variance. But as we don't know how many cows are there in each village in advance before taking a sample, so we don't know what is population variance also. So when the population variance is unknown, we use pilot surveys result. Small sample we will take and obtain SY square sample variance, and accordingly we decide our sample size. So for example, suppose population size is 1000 and D is 1. What does that say? Alpha is 0.05. What he is saying, user is saying that you do give me sample size in a such a manner that the population mean and sample mean absolute distance should be less than 1. Absolute distance should be less than 1 with probability 0.95. That means in 95% samples, all possible samples, your estimated value and true value, which is unknown to us, the difference should be less than 1. Difference should be less than 1. And when standard deviation of the study variable is 4, that is variance is 16. In this case, the minimum sample size using this formula uh, comes out to be 58 using this formula. And using this formula, for capital N is very large, assuming that the population size is very large, it is 62. Uh, more than 62 you won't need. Minimum 62 units are to be tested. Out of 1000 population, you must take at least 62 element in to ensure that the population mean and sample mean, the difference will be less than 1 with probability 0.95. Same thing, the standard deviation is same, but you are now allowing the more difference. If you are allowing more difference between sample mean and population mean, user is allowing more difference, double difference, then sample size significantly reduces. Then only 16 sample size, because you use this formula for D is equal to 2 now. So 16, it is 15 point something, and for larger n also it is comes out to be 16 only by putting value over here. Both are 15 point something, rounding to 16. So in this manner, you can see that the if your D is small, sample size is large. If D is small, and D is large as compared to SY, standard deviation. See, D is 1 here, also D is 1, but standard deviation is lesser here. Standard deviation is lesser, then sample size is 16. Sample size is 16. Even though here 1 4 is there, 58 sample size, but 1 2 standard lesser standard deviation. As you might be knowing, most of you, when you check whether the rice is cooked or not perfectly, you take only one piece of rice, that is sample size n is equal to 1. Why? Because you know that the variation in cooking is very less over there. So lesser the variability, lesser the sample size, and higher the variability, higher the sample size. Higher the variability, higher the sample size. For the same D, we are comparing 1 1 here and 4 2 here. This is your homework. You find out the for standard deviation is 5 and 95% confidence interval you are looking for. Then D is 1. Then what should be the sample size? Mini type demonstration I will show you after this proportion also. Determining the sample size required for proportion purpose also. So we have to solve this equation. Statistically, this equation is solved. What is the equation? Probability that estimated value of population proportion and true value of population proportion, which is unknown to us, the difference should be less than D with probability 1 minus alpha. In order to satisfy this equation, we need sample size like this. So whenever you are estimating proportion, you have to use the sample size formula, this one. What is N here? Population size. D, the difference you are allowing as a user. Z alpha by the, what is the level of significance you have taken, that alpha you have to declare accordingly. Z alpha, for example, alpha is 0.05, Z alpha by 2 is 1.96 everywhere. And P is the true value of population proportion. Now you may wonder, as you are going to estimate the P, then how do you know P in advance to find the formula for N? This is very interesting. Our problem is to estimate P itself. So how can you use P in the formula itself? So you cannot use. So for that purpose, you try to carry out small sample survey, pilot survey it is called. Take 20, 25 or 50 observations first of all, and try to estimate P. Uh, P is nothing but D divided by N, number of people in favor of that fellow divided by small n. That P you can estimate, and that you can put over here, and accordingly you get the minimum sample size. For example, you have taken 50 sample as in a pilot survey, and the sample size comes out to be 369 minimum sample size. So later on you can reduce 50 from here, and remaining elements you can take from the survey, your planned survey. Okay, capital N is 1000. In this formula you can put D 0.05. That, what does that say? He is saying that the difference between the proportion you are going to estimate and true value of population proportion, it should be less than 5, 0.05. That is, if the true proportion of people in favor of politician is suppose 45%, so your estimate should be in the interval 40% to 50%. He is saying like that, with probability 0.95. So what should be sample size? So you take a sample of 369 in that case, and if your capital N is very large, then this formula you have to use, that is 398. There won't be much difference here. Okay, so for example, you have 1 lakh people, 100,000 people in the population, and you want more accuracy. The difference between the true value and estimated value should be less than 0.02. That means only 2% plus minus you are allowing. With 95% confidence you want, and the proportion of people in favor of politician, pilot survey says it is 50%, suppose. So your sample size required is 2401, and for large N 2449. So please note that even though the proportion of people in favor of politician is 50% in this both example, but here he is allowing 10% plus minus error, then only 97 sample size is sufficient, and here he is allowing only 2% plus minus. So we need 2400 sample, or for large N, population size is very large, 2400, around 2500 people you have to sample. Suppose the cost of sampling for one person is 10 rupee, then here you need around 25,000 rupees, and here you need only 1000 rupee if your accuracy level desired is smaller, because D is larger means you are looking for lesser accuracy if D is large. If D is small, that means you are looking for higher accuracy. So For proportion, I have given formula here to while estimating proportion, how to decide sample size. And in last slide, while estimating population mean, many scientists, they need to estimate population mean. While deciding, accordingly you can decide your sample size and then conduct the experiment. Minitab demonstration, I will just show those who are familiar with Minitab. You can go through, this is Minitab software. You can go to stat, in this menu, stat, power and sample size, sample size for estimation purpose. And suppose you have a normal data and standard deviation, uh, let me share the Minitab with you. This is Minitab software. Here you have statistics. In this menu, stat, power and sample size. For sample size for estimation, you have to go here. And then mean standard deviation is suppose 1, and estimation of sample sizes you are looking for. Margin of error for confidence interval is suppose standard deviation is 4 and margin of error is 2, 2, and in option you have to give 95% two-sided. Here assume that population standard deviation is known and that is given to be 4, and margin of error D is equal to 2. Then let us check here. So the sample size comes out to be 16. Sample size comes out to be 16 here, when margin of error is 2. If you change the margin of error 1, check the sample size. Sample size 62, as we have seen. Here it assumes that the population is of large size. So in bracket, those values I have provided in my presentation, those are matching with here. Those who are, you can get familiar with this Minitab very easily and use this power and sample size, because it needs a lot of time to discuss discussing all this, because there are many issues here, Z-test, T-test, pair T-test for one-way ANOVA and others also. You can decide the sample sizes. For variance estimation purposes also you can decide sample sizes. So for example, in proportion case, we can go for the binomial proportion 0.5 is pilot survey says around 50% people are from politician side, and 0.1 10% plus minus you are ready to accept. Okay, 95% two-sided. Okay, and so the sample size comes out to be 104. I have written in that presentation 104. If you change the margin of error, for example, this is 0.02, 2% plus minus only. In that case, sample size will increase. That is 2,449. So from Minitab also you can use this directly, or you can use those formula which I have given. So a lot of applications you will find in this stat, power and sample size. There are for given a power, what should be sample size? These reverse problems, those who are more familiar with statistics, they can use this more effectively, this command, and get your work done here. So this is Minitab demonstration briefly I have given. Uh, let me finish here. Thank you for your attention. Uh, this was very small lecture and very brief lecture regarding the sample size determination. I hope you might have understood how does the standard deviation, increased standard deviation means increased sample size, lesser standard deviation, lesser sample size. Formula you can use, or you can use software Minitab also and get your work done. Thank you for your attention for this lecture.
Thank you,
If
sir.
you
We
have
are
any question or query, you can ask immediately now also.
Yes, sir. Thank you for your kind of insights towards the topic sample size. And I think we got to learn many things from here in this lecture. Definitely, it's a very vast subject which cannot be dealt in a stipulated time as well. So, now moving towards our question answer session, which will be taken over by Dr. Pratima Pawar, ma'am. So, Dr. Pratima Pawar, over to you. You can take over the question answer session.
Thank you, ma'am. I am obliged to having this opportunity of taking the question answer session. I am thankful to the organizers for giving me this opportunity. And sir, your presentation has covered all the aspects of the sample population, its size, its mean, its confidence intervals. Very lucid and fluent explanation with deep knowledge, sir. So thank you so much. There are very less questions, you can say. The question asked is, which sample procedure will be used in the social science studies? Sampling procedure.
Yeah, uh, we need separate lectures on sample theory, as I give main research methodology, 2-3 lectures on sampling theory, because here it was only one lecture and I have to cover many issues. Anyway, in sample social sciences, we deal with the population of homogeneous nature also, and sometimes heterogeneous nature also. Homogeneous nature means almost there is less variability in study variable. For example, the study variable is the size of the household. How many people are staying in a one house? That is a size of the household. So this variable may be homogeneous in the same population. Suppose there are 1,000 household in a village, and this is the study variable. How many people are staying in that house? That is, in this regard, if this is a variable size of the household, then population is homogeneous. And if you see the another variable, what is the income of the household, then there may be some rich people in out of those 1,000 families, and some of them may be very poor, some of them may be middle class. So in that case, we have to use stratified sampling. If your study variable is just to estimate the population mean of the income, average income, if you want to estimate, use stratified sampling. If you want to estimate average size of the household, then you may use the simple random sampling only, considering the population is homogeneous. Homogeneous population, simple random sampling works well, and for heterogeneous population, you may have to go for the stratified random sampling. And how to allocate the sample sizes in each strata, there is a allocation problem also, stratified sampling takes up. Okay, any other? You can
Yes,
use
sir. If we
cluster
are
sampling
using,
and two-stage sampling, multi-stage sampling. There are many sampling procedures for social sciences. Multi-state sampling is also more useful. First of all, you select districts, then we select villages, then we select household. This is multi-state sampling.
sir,
So
if we are using only one criteria, for example, the water used in the household by each family. So
yeah,
what type of sampling will use?
here amount of water used by family, instead of this I will be more interested in using estimating average amount of water per person.
Per person.
Average amount of water per person, this might be my interest to estimate. So in that case, I have to estimate, I have to decide first of all the list of the population element. What are the population elements here? Household is a population list of household. From where we get from corporation or villages. You get the list first of all. You select random sample and choose those, visit those households only for urban area and for rural area. If you see the urban area, people are using larger amount of water as compared to rural area people. So that might differ. So your classification may be urban rural, or in city also, slum area, and some sophisticated areas where people are using more water. So if you have some background knowledge, so you have to stratify your population accordingly.
Thank you so much, sir, for your nice explanation. And over to you, Alia Naz, ma'am.
Thank you, ma'am. And I think sir has explained us and given us lot of things to understand in a brighter way and to know the concepts in much more vibrant way. Thank you so much for your expertise, sir, and I really uh, become humbled here to thank you once again on behalf of the organizers as well. Now moving
Thank
towards
you,
our
madam.
yeah, moving towards our second technical session of the day is regarding teaching and participatory and innovative learning. A teacher is a learner. Until he is not a learner, he cannot become a teacher. So, a teacher teaches, a good teacher explains, and a great teacher inspires. So, we always should strive to at least inspire one person in our life, as that will be our biggest award as a teacher. So, with this thought, I would be welcoming our next speaker of the day, and he is Dr. Subroto Hathi, Assistant Professor, Department Dairy Microbiology, SMC College of Dairy Science and Agriculture University, Anand, Gujarat. I welcome you on board, sir. Now, I would like Amol Jadavji to give a brief profile of our speaker to the participants. Over to you, Amol.
Thank you, ma'am. Good evening, everyone. I extend a warm welcome to all on Faculty Development Program. It is a great honor for me to introduce and welcome our speaker of this evening, Dr. Subrata Hathi, sir. At present, Dr. Hathi, sir, is working as Assistant Professor in the Department of Dairy Microbiology, SMC College of Dairy Science, Anand Agricultural University, Anand, Gujarat, India since 2012. Dr. Hathi sir published more than 70 research papers in reputed journals nationally and internationally, 8 review papers and 3 edited books. He is the secretary of Swedish South Asian Network Fermented Foods, Anand, and executive editor of International Journal of Fermented Foods. He is also the editorial board member of Letters in the Proteins and Peptides. He also handled the externally funded project sponsored by DST, DBT, ICAR, Government of Gujarat as principal investigator or co-principal investigator. Dr. Hathi sir is a recipient of numerous prestigious awards, which include NAS Young Scientist Award sponsored by NAS, New Delhi. Professor Sukumar De Gold Medal from WBAFS, Kolkata as a university topper. Urmilabai Gold Medal from Indian Dairy Association, New Delhi. Early Career Research Award by DST, New Delhi. IACBG Fellow by DBT, New Delhi. INSA Visiting Scientist Award in 2018 by INSA, New Delhi. Best Teacher Award in 2018 by ICAR and Anand Agricultural University. And also, He is a recipient of Dr. Ms. Gauri Ganguly Memorial Award in 2019 by Indian Science Congress Association, Kolkata, and also visited the University of Copenhagen, Denmark, for presenting the research work under Indo-Danish collaborative workshop in 2018. He is also the elected fellow member of National Academy of Sciences, India. We are really honored by your presence in this FDP, sir. Thank you, sir. Now I hand over to you, sir. Thank you.
Thank you, Dr. Amal. Am I audible now?
Yes, sir. Yes, sir.
Okay. So very good evening to all of you. First of all, I want to thanks the International Journal of Microbial Science for inviting me to speak a very extremely important topic is teaching and participatory and innovative learning under the 1 week faculty development program on statistical analysis in academics and research. So teaching and participatory and innovative learning, you see that these three important components are very much important for us as a teacher. We are every day facing our student in the class, and they are mostly dependent on us. So we are making them attracted, means we are making them involved in our teaching systems, so that there is a two-way exchange of the knowledge. So I will take you to the different path, that how can we teach in a better way for the students, and how we can also enrich our knowledge in the class, and even the objective of this 1 week faculty development program is the how you can increase the API score also from this topic. So when this topic, I have seen that I have to talk about it, so I was a bit uh, thinking that how can I start the topic. So I was finding that is there any definition on teacher. So I got that the UNESCO has given a definition that teachers are one of the most influential and powerful uh, forces for equity, access, and quality in education, and key to the sustainable global development. So you can see the influence, you can see the inspiring capacity, you can see the motivational force of a teacher to develop the entire nation by offering, by contributing a good citizen to the society. So this is the definition of the UNESCO, that the whole entire world can change by a good teachers. Just Alia Naj ma'am says that teachers are good when they really shows the path through which the students can walk. So whenever we talk about the teachers, we always remember our legendary peoples who really is the best teacher in our culture. One is Swami Vivekananda. He used to say that arise, awake, and do not stop until the goal is reached. So you should always inspire your students. They may get failed, they may have some difficulty, they may face the obstructions, but don't let them go away. Don't give up. Every end there is a new phase of life. So you start, you involve your students, you inspire your students, so that they can go ahead. And always we remember the Teachers' Day on 5th September every year at the birth of Sri Sarvapalli Radhakrishnan. He used to say that teachers should be the best minds in the country, because their mind means teaching is a very noble profession. You cannot estimate but just by quantity like money and everything. When you will teach in the class, you need a very good preparation, because you are throwing something which your students are waiting for you. So teachers should be the best minds in the country. Then only you can give something, because in our class, always the teacher is the only persons who never expect anything from the students. You always in the position to give. So the teachers should be in the mood that they can deliver the best. And whenever we talk about the teacher and the gurus, we always take the name of Dr. APJ Abdul Kalam. He is our modern teacher, you can say. Teaching is a very noble profession, he used to say, that shapes the character, caliber, and future of an individual. If the people remember me, means the Dr. Abdul Kalam, as a good teacher, that will be the biggest honor for me. So you see that how he was proud for his teaching ability. He, whenever he goes, mostly he visit to the schools and colleges and the universities, and he used to ask the students to, they have any kind of questions they used to ask, and he said that there is no any kind of questions which is very formal. You ask what is in your mind. So he always give the preference to the students to open up your horizon and come ahead. So now come back to the our system means university or college systems, where we used to teach the UG, undergraduate, postgraduate, and PhD students. So we take the theoretical teaching class, we take the practical classes in the laboratory. We also make our students to visit the library, to read some good books, to write, to prepare a notes or assignments in the library, because library is the seep of knowledge, all of you know. And we used to take the student outside the class in educational visit, and also we encourage the students to take part or to participate in the extracurricular activities, to explore their hidden talents, literary events, songs, fine arts, which whatever they have in their minds, they can participate, they can explore, because all of means each individual has their own hidden talent, so they have to explore it. So we have to give them some space even after the regular curriculum, so that they can explore their own findings also. And always the students should be also make them mentally and physically fit by doing exercise and yoga, everything. So it is now the part of the university systems. Now coming to the theoretical class, theoretical teaching class in the university or college systems. So when we'll take the class, basically the teachers have some responsibility. Teacher must have the proper preparation with notes, updated knowledge, and full confidence before taking the lectures. So this is a very important part. If you have a class tomorrow, you must have the proper, what we can say, that proper rehearsal before taking the class. It will give you better confidence, even though you are taking the class last 10 years, last 15 years, but you just update your knowledge by reading good books, by reading good review papers. All are available in your systems, in your internet, and you can update your students about the good knowledge. But another important point is that when you will enter the class, the students are always wait for their lectures. So when you will start a lecture, don't start that by just doing a attendance or any any very normal things. You start the lecture with a short relevant storytelling mode. You start that today morning I read a newspaper and I was so surprised that this fellow has done remarkably good things, because he did very good efforts in his sports or in his chess. So whatever the story you can share with them, then what will happen? The minds of the young students will come to your place. They feel comfortable. They will be more attracted to you. And the third point is, it is a very well-known fact that when you will teach a class, you follow walk, talk, and chalk. Walk, talk, and change chalk means you don't stand in a side. You move around the students, so students will follow you. And secondly, you talk just facing them, not talk in a way that they are not seeing you, and always write something on the board. So this is the best method for better teaching. But nowadays we mostly have we have a good option of this is the multimedia systems. Then use simple language, if required speak in Hindi or any regional language, so that when you think of you are explaining the transcription or translation or protein synthesis or gene expression, you don't directly go ahead in the hardcore microbiology. You just start with a very simple language. If it is possible, you can speak in the regional language. That will help them to understand, because if they are not getting interest on your topic, they may not listen you properly. Maybe they are they are physically present in front of you, but they won't take the interest. So first you make interested between you and your students. Ask questions while teaching, because it will say that the students are listening properly. Tell anyone to brief the entire talk at the end of the class. Basically, I follow these things when the class is over. So it is a narrow 55 minutes class. So after the class, I try to finish within 45 minutes, and then I call any one of them, or first I allow them to, you come and speak what I taught today, just 40 minutes ago. And if they are really interested, they will come and they will start to speak. When they will start to speak, they will not completely say everything, but you just help them. You just help them to speak, to make a good story, to make step-by-step process. You can see the interest of the students will be quite high. You can't even expect. Then theoretical teaching or in classroom, there another points are that take help of smart features, because I've already told that nowadays all the classes we have a good multimedia systems. Keep surprise tests to evaluate them. Don't inform them. You can, but that is for their interest. That is you can also evaluate that the method you are teaching, really they are grasping you, really all the materials you have taught today has been transferred their minds. So you can keep one surprise test, very simple test you can keep, and see the evaluation of you also, of the teachers also, evaluation systems. Always try to give the example from day-to-day life. But I said that when you explain anything, try to take the example from very simple way so that they can understand well. Always encourage them to ask questions. You tell them that when I will speak, when I will teach you anything, if you are not able to understand, you just hand and ask, repeat. Sir, can you repeat again? So again I can repeat. So that is the things that the students are taking interest. And also encourage them to initiate the startup. So startup in the sense that the some of the topics which are having a very practical aspect. So you can tell them that these are the practical aspects. You can also think about it, and you can even take the idea to a better way, and start your business even. Sorry. Then theoretical classes, we have to give them a class talk on recent happening. Just you can see the coronavirus. So you can give them one topic, and tell them, you can say a few words about the coronavirus. So that will make them confident, that it will also help them their communication skills. Make the class interactive from both the sides. So your class should be from a very what can I say, both way exchange. Means I can take one example. One of the class in my class, one of the student asked me one questions, and I did not know the answer exactly. So what I said, I don't know the answer, but your question is quite interesting, and I will definitely answer in the next class. So I'll again read it, I'll find the answers, and tomorrow in the next class I will explain it. So you see that the both way we are get benefited. Not that the teachers knows everything. It's not possible at all, because the mostly the life science or biological sciences are changing in a very faster space. So you have to make you updated, and students are very intelligent fellows. They really read all these things, and they make them updated, because they are trying for different competitive exams in the coming years. So they are reading all these things. And another thing is that keep a good relation with students. I'm not that you make any very close relations. You can make them comfortable in the class. When you will enter the class, it's not that they feel a bit awkward, or they feel a bit fear that this teacher is very rude in nature. So you just make them communicate in such a way that they feel comfortable, and then you can see that the whole class will get changed, and you will have a very vibrant class. If need extra help, take extra classes. What in our systems we do it. Some of the students we know, they, because all the students cannot have the same frequency to learn everything. So you can ask them. In our system we have already started that those who are really having the problem, because after taking one exams, we can find out that some of the students are having these problems. So we call them after 5 o'clock, if if they don't have any classes, and they can come and they can say that these are the problems we are finding. First we tell them that in your hostel, because most of the students are staying in our hostel, so you can take the exam, you can take the help of your friends, because they will make you more understandable. And then if it is not solved, you must come to us. And they used to come, 2 3 students in every semester, they used to come. So this is the process. If you start, and I think the students will get benefited, and in future they will remember you. Maintain punctuality, discipline, decorum in the class from both the sides. This is very important. When the knowledge is exchanged, when the science is being taught, it's not a child play. It's really a very vibrant or very calm and quiet place is required. Then only the same frequency will get matched, and you will get a resonance out of student-centric teaching methodology. When you will start a teaching, your focus is that how can I make our students benefited. So open-ended projects, give you open-ended projects, passion projects via DIY labs, classroom collaboration and team building, make a group and give them some problems so they will solve it. Implementing creative arts sets in a new life. Journaling or enhances their creativity. Brainstorming session, flip at the best, means the homework will be done in the class and classwork will be done in the home. So flip side systems, unconventional learning materials, TED talks. You can talk them any topics, so they feel that it is the out of syllabus, they will take more interest. So in this way you can make them interested in other way. Field works, you take them to a visit to the field, means in laboratory, another way, and show them all these things. Utilize the visualization, as all of us using the PPT and other things. Use simulation, MR, AR, means audio visual, everything you can use it to make them benefited and explain these things in a very simpler way. Tools of active and innovative learning. So when the innovative learning we talk about, there should be interactive and participative approach from both the sides. So Philip method I've already told. Cooperative means collaborative, or make a group, give them a project, and also you can say that we can have a group discussions in a 4 5 students in a group, and they can discuss in one topic. So both of way they can get benefited. Problem based learning, active lab classes by learning theoretical concept, learning by doing. So some of the theoretical we say, but we cannot make them understand understood properly. So what we can do, if possible, we can arrange these things in a lab, and tell them they can do it in a practically, so that they will understand all these things. Also they can use the different open sources. Open sources means the e-courses which are available and which are already available by UGC, like SWAYAM. In ICR system we have a SERA. So e-courses are already developed for UG and PG. So they can also share the open sources for updating their knowledge, and you can help them. Then comes to the practical classes in the laboratory. So always engage them in the practical classes. Maybe your classes is a bigger, more than 50 students, but make a group of 6 students or 7 students and give one particular experiment, and let them do in hands-on practice. Properly demonstrate and simplify the experiment. So before starting, you must demonstrate, or you must explain that how you can do this practice. Explore principles through concrete or simplified examples. Establish the hypothesis or theoretical concepts through the experiments, and develop their technical, observational, motor skills to become an independent thinker. Because the practical class mostly increase their analytical brain. So you make them encouraged, you make them engaged in every practical class, and let them do in hands-on systems, not just by demonstrating it. Don't do it. You arrange some good experimental accessories, and let them encouraged, let them engaged. Then only they will learn something. Encourage them to classify the data, explain results and prediction. The Means when they will do the experiments, after end of the experiment they will get the results. You first tell them to explain their interpretation of their own experiments, and if they are not able to, you just make them correct it, but don't tell these things, because in your microbiology what we say that if they do the plating today, the result will come after 24 hours. So they have to come here in the laboratory, and we give them 1 hour time to write the interpretation in front of us by seeing their own experiment. So in this way they can write the interpretation, they can uh, make the table of the data, they can think of that how the standards are being formulated. So make them encourage, make them engage, see their own experiments, how they can interpret the results. Work with other students, which can lead to a greater motivation toward an interest in the subject. So make a group, don't give one experiment to one students only. In the exam it is okay, but when the day-to-day practical class, at least 2 3 students you can make in one group, and give them one experiment and they do it in a proper way. Next is days, because most of the students I have seen, they are busy with the smartphone. So from the day one when they will enter in your campus, means the newly admitted students, you encourage them to visit library 3 to 4 days a week. At the very first day when you will face the students, newly admitted students, fresher students, you encourage them to visit the library, read any books of their choice. Don't ask them to read only microbiology books, only biology books. They can choose any of the books, but make them habited to visit the library. Prepare note or assignment. You give one assignment and tell them you go to the library and find books, make a bibliography at the end of the assignment, so that you can see that these books are available in the library, and they have read it and they made a very good assignment. Suggest, you can suggest some name of the books or new editions every year for updating their knowledge. In our campus at Anand Agriculture University, we have a very good library facility, and library is open from 9 a.m. to 11 p.m. in the night. So and library is very nearer to their hostels. So after having a dinner, they can go to the library at least 1 or 2 hours, and they can read over there, they can make their own notes, they can study anything. And even our library is also open in Saturday and Sunday. So students has a very good scope to spend most of the times to the library, because library, I already told, it is the seat of knowledge. Now comes to the extra supports to the students, because in daily day-to-day class or day-to-day course curriculum, we have to encourage them to participate in the extracurricular. I've given one very beautiful picture here. You can see that PV Sindhu and his coach Ulalam. So PV Sindhu and his coach are giving a good efforts, not a good efforts, I can say it's the best efforts they have given. Then only PV Sindhu has risen in the world ranking number. So as the students has the responsibility, even as a teacher, we should also encourage them. We should have to give them support. We should have to give them extra supports, so that they can grow well. So in my university, I have been working as the ex officio advisor for the Students Representative Council in Fine Arts. So those students who are really interested in fine arts, they can come to me, and we have a good fine arts competition in our college. So we organize it. I am also the ex officio advisor in the same SRC, means Students Representative Council, for the college magazine. Every year we publish it, and I also acting as a mentor for guiding the startup grant project of Miss Upasana Nath on ready mix probiotic fermented beverage sponsored by government. So we also give them mentorship to the startup. I'm also acting as the editor of DSC Newslink of our alumni association, and I am also acted as a net coordinator of dairy microbiology subject for ICER net. I give coaching to our students who are studying MTech second year, and they will appear for the coming December for the net. So Saturday and Sunday we used to give them the net coaching, but in nowadays Corona there, so we are taking the classes on online mode. Part of anti-ragging monitoring cell for first year BTech and MTech DT. So I am also the involved. And when they enter into the hostels, because you know that the UG students, they have just come out from their house after completing 10+2. So we make them comfortable in the hostel so that there will not be any kind of panic after coming out of their house. And I also invited in different delivered lectures. I did give the delivered lectures in training come coaching for ICRNet or dairy science students. And I also take the students to the outside to the different plants or industries where they visit and they learn something from their course curriculum. And I do the counseling for the extra counseling for the students who needs the extra supports. And I also help them who are preparing for different competitive exams like CSRNet, ICRGRF, ICRPG exams. And I have given two very beautiful pictures here that it is Mr. Sachin Tendulkar, our hero in the uh, cricket sports. So it is his coach. So you just see that at the last day of his coach, he is taking with his last intimations. So that is the closeness of a teacher. This is the bonding of a teacher and students. They never forget it. They will remember, because when you will support them, when you will help them to grow, they will never forget you. It is the ever bonding between teachers and students. Also help the student how they will study, how they will make the study plan before the exams. You tell them that you follow the lectures note which you have taught in the class of UG courses, online books and hard copies of books available in the library. You visit, there is a lot of books, you read it. You also update your knowledge by just reading the review papers. Note down each points and data with ear, because most of the competitive exams they follow the multiple choice question MCQ. So they usually ask different data, so you have to remember it. So you just note down it. And available e-courses for different UG and PG courses, you must follow these e-courses. I also advise the students, what you read you must write. Writing in one piece of paper the gist of what you read, it gives immense helps when you will appear in any exam. While going through the question paper, you may see the image flashback of the thing you wrote yourself on the piece of paper. You will see that the questions which are being asked in the exam, it is for you or for me only. So that kind of confidence you will get if you write after reading entire syllabus or the important questions. I already told that uh, students, those who are appearing in different competitive exams, they can make a group discussions, because make a group of 3 or 4 students help them to clear their concept. Assign a particular topic to students, prepare well and discuss only the facts and figures one by one, and listen carefully. In group discussion, you make them habit that first to listen what your friends are telling, then you react. Don't interfere in between their talkings or they are telling any facts. So make them a good environment so that they can discuss their own topic in a different way. And also help them how they can crack the exam. So give some mantra. It's not a mantra exactly, some advice, some what can I successful advice to the students. First tell them, when you will reach the exam center, you sit over there very calmly, and first when you will get the question papers, you must read it very carefully. Don't think that I got the question because of what I know, I have just started. No, you first read it very carefully from the first question to the last questions. At least give 5 minutes for reading the question papers. Leave tough ones for attempting the later. So if you think that the students think that this is the bit tougher, so first what you know those questions, you just attempt them fast, and then you attempt the harder. Identify terminology meaning of what would help in recognizing the answer, because always most of the competitive exams are MCQ, so there are some terminology will be there. So you just try to remember the terminology. You read the terminology repeatedly and you can able to remind it. Answer it in your mind fast, because in MCQ there are 4 options. So which is coming first after reading the questions carefully, you take that one, select the best answer and read every answer. It's not that you won't read only just C or B you are thinking, and you are abruptly you are taking the right answers. No. Answer the questions you know first. Make an educated guess. If guess is there, don't that give we take the name of God and make any correct answers. No, you just try to correlate the other options, and which is the best one, you just take it or choose for the correct answer. Secrets to conquer exams. How will you succeed in an exam in a successfully? Start early. If your exam is in the 10 o'clock in the morning, you must reach the exam center at least half an hour before 9:30. Time yourself. Don't be hurried. The examiner already told you have a 3 hours exam or 2 hours exam, so you have a time, so you manage accordingly. Revise and revise. If you have finished all the answers, you have a time another 10-15 minutes, you revise and re-revise it, because you will find some wrong things and make it corrected, and you'll have a better of marks or better score. Discuss with peers what you learned. So if you have any difficulty, you have not your concept is not clear, so before the exam you must ask those who really know the actual concept and make it clear, make the concept clear. Stay calm and composed when you will in the exam hall. Be very calm and don't talk to anybody. Don't make any hurriedness or any kind of things, because your brain has to work now, so you have to give ample scope the brain to work for you. Don't worry about level of competitions. You don't think that I am attempting the IIT level exam, I am attempting the CSI net, I may not get succeed. Don't do it. Don't think it. You think that it is my duty to give the best, and you will do succeed. Sleep well. This is very important. Before, one day before, if you have an exam tomorrow, you must sleep well. Don't think that what I know what I will do in the exam. Don't do it. You must sleep well. So now I have given a very good idea of how will you take the classes in the UG, PG and PhD, and how will help the students to grow well. Now coming to the innovative teaching methodology. What are the innovative modes? First is the voice thread. Voice thread is to build student engagement. It is basically is a collaborative multimedia slideshow that holds the images, documents and videos and allows the people to navigate the slides and leave communication in different ways using voice, text, audio file, and video. So voice thread is a web service that allows users to upload the PowerPoint slides, videos, photos, and you can ask the questions immediately or even after the video is being uploaded, after the video is loaded, and then you are asking, you can able to do it. You can do your voice recorded. You can ask the question directly by calling them, or you can ask in the chat box. You can ask the questions also. So this is the very good one multimedia systems, online systems, where you can use it in your effective teaching methodology. Another is the blogging. Blogging is a public post. Student can post their case studies in a class blog. Student can be asked to post note on the class blog and can analyze, evaluate and create the material. Blogging causes you to reflect. Teachers naturally think back on what has happened in the classroom and often wonder what they could have better done. Means the teacher also can evaluate their class, just seeing the feedback from the students in the blog. And even the student can upload their assignments in the blog. Blogging can help with the process, enabling teachers to keep ongoing personal record of their actions, decisions through processes and success and failures. So blog is a good way to communicate between the students and teachers by just following their feedback. Then another important innovative teaching method is Prezi. Prezi is an Hungarian presentation software company that is founded in 2009. And as of April 2018, Prezi had more than 100 million users, you can imagine, who had created more than 325 million public presentation, and that have been viewed over 3.5 billion times. Prezi is a versatile app that lets you make professional looking presentations. It's like a free pared down version of the PowerPoint, and Prezi lets you make presentation that are as casual or as a professional you want them to be. It allows you to add information to Prezi, organize it in a logical way, embellish it with audio, video, and then share it with the people you want to share with them. Prezi makes exactly the presentation very easy. So it is one kind of PowerPoint making tool where you can very effectively you can design, you can add your voice, and you make a very good presentation with the help of the software Prezi. Another one is the screencast. Screencast is a digital video and audio recording of what occurs on a presenter's computer screen, and it can be used to create sophisticated, information-rich multimedia presentation. The word screencast was first coined by the columnist John Udell in 2005, and screencasts are an effective way to share ideas, deliver content, obtain the students' feedback. Screencast can be used for describing a step-by-step process, explaining a particular concept, or presenting a PowerPoint presentation with the narration and multimedia elements. So there are some of the freely available softwares of the screencast is Zing, Screenjelly, Screencast. So you can use it. You can make a very good PowerPoint presentation of your talk, and you can share with your students, and even students can also share their ideas, innovative ideas through this screencast. And another important innovative teaching tool is the social media. Nowadays, all of we know the social media means Facebook, YouTube, blogs, Twitter. So social media where individuals are in a communities that share their ideas, their interests, their happenings. So you can also use it in for educational purpose, in teaching purpose. So you can put one assignment in the you can share your student with your students, or you can ask one idea that I'm thinking these things, really it works. The students will give the feedback. Maybe they are undergraduate or postgraduate students, so they will give you the feedback, and both way you will get benefited. Another is smart board. Most of us, we use the smart boards in our class. Smart boards transform your learning space into the interactive, collaborative and environments where students are both inspired and focused. And with a smart board, interactive whiteboard, instruction can enter a dynamic multimedia content and write notes in digital ink, then save them instantly and distribute this material to students with ease. Smart board interactive whiteboards make learning a visual, engaging experience for students, which also helps deepen understanding and promote the retention of course material. Means smart board means, you know, that a very complicated figures, just I can talk about the DNA replication process where a lot of enzymes proteins are coming in a step-by-step manner. So if possible, you can ask your students and you can tell them that can you name this protein. So you just make them underlined, and they will say, they will find, or you can just tell them this, find out this DNA polymerase 3 or DNA polymerase 1 from this figure. So if really they have attended the class, you call them and tell them to find out in the smart board, they will find it out. So smart board is one kind of a interactive board. Then open system, open source system means e-courses. So Moodle is the virtual learning environment which provides staff and students with access to electronic teaching and learning materials such as lectures, notes, and links to the useful website and activities such as discussion forums or groups. It is something that lets you capture your experience, note, website, and so the government are really taking good initiatives to create lot of open source systems. Even all the journals, all the books, which are really very important books, now most of the universities are sharing through the different online systems. So the students, you encourage your students to download those books, download those notes, or the courses, e-courses, so that will help them to make their good notes for cracking the exam, for cracking the national level exams, and enrich their own knowledge. So student learning methods, I am coming just end of our entire presentation, that when you will teach the students, what are the things you should remember. The presentation open with question and answers, real-time project works or field works, that is most effective way. You take them to the field and show them, and that will give them a better way to think about. They can give you better way, in an innovative way, the answers. So that is only possible when you will take them to the fields, means where the practical things are going on, or make them practically involved in the field, so that their knowledge, their skills, and their ideas, nowadays idea is very important. So reflective papers, reviews on paper or book. So I tell them, I always used to tell them that if you want to update your knowledge, you read the good books, you read good reviews which is published by very good journals, and update your knowledge. rigorous analysis and synthesis. So you evaluate a student. Just I have taken one example of the surprise test. Identification of problem, in-depth case study. Give them a particular problem and tell them how to do it, but don't tell them how to interpret it. So that's where their depth of knowledge will increase. Multidisciplinary projects. Don't give only microbiological work. Give the project work in such a way that they have to take the help of chemistry people, they have to take the help of management people, they have to take the people of engineering people, so that their knowledge level will be knowledge quantity will get higher, and their skill will get again encouraged or intensified. Use gamification, or means fun embedded means of TLP, means the things which they are doing in the practical classes, that is not monotonous. At least they can finding something, that experiments are giving them a better interpretation what exactly they are finding. So this way you make the class more interesting, because if the students are not taking the interest, it will be burden for them. And very common things we usually follow in the class teachings are the chat rooms, discussion board, webinars. Nowadays it is a best tool for us to connect it with our students by taking the classes through different electronic platforms like Zoom or Google Meet or other things. We can also communicate with them emails, and we also use the WhatsApp and Facebook and other things for the teaching purposes. And image creators, we also tell them, we also send them some good images of the particular biochemical reaction, so that that will make them clear in their so that it will clear their concept. Now I'm coming to the last slide. It is the last slide of my presentations, because FDP has another important purpose to how the participants who are mostly the participants are mostly from colleges, universities. They are the teachers, they are the students of PG, PhD, and postdoctoral. So they will appear for different exams for selection in the assistant professor or different scales. So I talk about the teaching, learning, and the innovative learning and the participatory systems. So it is the category, it is coming under category 1, teaching, learning, and evaluation related activities, where you have to, the marks is being already being given for a particular aspects, like lectures, seminars, tutorial, practical, and contact hours undertaken as a percentage of lectures allotted is for maximum 50 points. So your daily UG, PG, PhD lectures, you are attending some seminar, you're organizing some seminar. Organizing is different part, and practical classes. So contact hours, as per the contact hours, your points will be different. And as per UGC, your contact hours minimum should be like that. Lecture or other teaching duties in excess of the UGC norms means you are helping your students by just counseling or other things. So it will have another points for you. Preparation and imparting knowledge on instruction as per the curriculum. You are following the syllabus by providing additional resources to students. You give them a notes, give them a PPT, or you have written a good books you can offer them. Use of participatory and innovative teaching learning methodologies, updating subject content, course improvement, examination duties, just like invigilation, question paper setting, evaluation of answer scripts as per the allotment of or duties. So these are different components under this. Mostly, I think that this part, there is no problem of any teachers to have this have the mark to get a full marks of it, because these duties are very nominal to, and it is assigned to most of the colleges teachers, because these are the day-to-day work. So in API score for teaching and learning and evaluated activities, there will be no there will be any kind of difficulty for the participants, and I think they will get the full marks. There will be no any kind of problems for them to have the good marks from this teaching, learning, and innovative. These are some achievements of my students who worked under me in masters and for PhD. They have got best poster award. They got the startup grant from the government of Gujarat, and most of the places my students had presented the research on, they got selected in different seminar and conferences. So that boost our confidence, because our students are representing us in different aspects or different fields. And again, I'll tell you that I have written these 3 books which has been shown here for my UG, PG and PhD. Recently I have published in Apple Academic Press with CRC in 2020. It is available in the CRC website. And 2018 I have been selected the best teacher award for by our UG students, and I have awarded with the best teacher award from our then governor Sri OP Kohli, government of Gujarat, in our 16 convocations. And now I am concluding my talk here just by saying a very interesting quote given by Mahatma Gandhi: "The best way to find yourself is to lose yourself in the service of others." That is the best way. He explained how the teachers should be for the society, how the teachers should be for the students. So now I am very much thankful for the organizers, the participants, and all the people who are listening me. Thanks a lot for listening patiently. Thank you all.
Indeed, it was splendid, sir, because as teachers, we also have to be updated with the latest technology available. And your presentation was apt for the time, because we are going through this pandemic COVID-19 situation, and these are really a kind of learning times for all of us. So with this thought, and now we are open for question answer session, and I would now over to you, Pratima Pawar ma'am, for the question answer session.
Thank you, ma'am, for giving this opportunity. All of us do not have equal talents, but all of us have equal opportunity to develop our talents. I thank International Journal of Microbial Science for giving us this opportunity.
Pratima ma'am, your voice is breaking. Your voice is breaking, Pratima ma'am.
Yes, ma'am. Yes. Is it okay now?
Yeah,
Yes,
now
yes.
it's okay. You can carry on, ma'am.
Okay. All of us do not have equal talents, but all of us have equal opportunities to develop our talents. It is rightly said by Dr. APJ Abdul Kalam. An International Journal of Microbial Science has given us this opportunity to develop our academic talents by organizing such a wonderful workshop FDP program for 7 days. I thank you, sir, for your simple language and very nice explanation and good presentation. There's a question. Sir, can you please explain the flip method in detail with example?
Yes, yes. Flip method, basically it is followed in the school systems. In the schools, what exactly happens that we give them some homework to do in the house. But what happens that the some of the students who are really interested, they do it, but most of the students, they do not do it. Okay. So what the teachers are doing, that those which are really important and giving them the homework, that will be done in the class, and in the class which they are usually teach them, that they are giving to finish in the house. So it is basically a flip system. Means what they are doing in the house, that will be in the class. So make them free from the education in the house. They should enjoy in the house. Don't be panic with the homework.
Okay, sir. Okay, sir. And brainstorming sessions, sir. Can you give an example of some brainstorming session which you use for your students?
Brainstorming session, it depends on the topic, just like COVID-19 now. They are very interested that
Okay,
what
sir.
exactly happens, why they are not allowing us to join the colleges. So you may involve them just giving this kind of topic, and encourage, means make the brainstorm about this. So brainstorming means it's not that the hammering. Brainstorming means the topic is so interested to them that the brain is really interested to listening.
Yes. Okay. Thank you, sir, for your nice explanation.
Thank you all.
Okay. And over to Alianas, ma'am.
Yes, sir. It was indeed pleasure listening to you and coming to know so many things about tools to be used alongside. Especially the blogger idea is spectacular, because we somehow as teachers, when we are working, we tend to underestimate the power of technology,
Yes.
whereas our students are digital natives, and we have to match to their levels to imbibe those knowledges and the knowledge what we have gained. Because of this online presence, most of the students take for granted knowledge is information, so which is not correct. So we have to gear up for that. So here we have George Couros, educator and author of The Innovator's Mindset, who says technology will never replace great teachers, but
Yeah.
technology in the hands of great teachers is transformational. We know this because we see that having access to so many different types of data on student performance enhances the relationships between us and the students. So with this thought, I expect that all the participants will definitely utilize all these points, whatever you have shared along with us, sir. Now we come to an end of the FDP, of the 7 days FDP, which we were going through on this 7 days. And any good start, any new beginning has to come to an end. And so is our FDP as well. So before we wind up our FDP, we would like to have a validatory function as well. So we have our chief guest for the day, Dr. H.R. Kulkarni, Principal, Jayavant Rao Sawant College of Commerce and Science, Harapsar, Pune. I hope he will be giving us more insights on how to continue further. I welcome you on, sir, and extend my warm welcome to you. And I hope all the participants will also bear with us for this validatory function. Over to you, sir.
Good evening, all. Am I audible there?
Yes, sir, you are audible.
Okay. Good evening to all. I'm in the beginning. I'm very much thankful to International Journal of Microbial Science, its editor, Professor Rajesh Dhakne, and the organizers, Honorable Shankarrao Gadhave and Dr. Sanket Doshi, who have organized this nice function on this nice topic. And I think so delegates might have benefited over the period of these 7 days. The speakers from different part of world were called to deliver their thoughts on this nice topic. And friends, the importance of measurement is in the research of field is quite different. In the late 19th century, where the tools for the measurement and accuracy were not so developed, the results drawn by one were challenged, and one were one was not confident regarding the result drawn by him. Measurement is one of the more important tool in the field of research. One researcher, when has performed his experiment, it was very difficult to repeat and test it in the different part of world, as the measurement methods of measurements were different at different places, different countries. For example, British and British ruled countries were using foot, pound, and second system for the measurement, where the other world was using their traditional tools for the measurement. The results in the other part of the world were not tested accurately, and it was very difficult to repeat the experimental procedure. Accuracy increased in the late 19th century and over the period of 20th century. The results drawn by the earlier researchers were tested by the next researchers with modified method of analysis and with precision. We have seen many principles and laws in the science have been re-explained by the new researchers with more accuracy. For example, the Newton's measurement of sound by Newton. The method was modified by Laplace. All the gas laws, where the van der Waals gas equation is nothing but the correction over the earlier gas equation. Various statistical and mathematical methods have been developed. Nowadays, the computerized analysis is also possible and gives more accurate result. The instrument with more accuracy and precision have been developed. This leads to get more accurate results where one cannot be challenged. The world of communication, industrial development, development in research and development field, space, medicine, agricultural, transportation, education. I am giving few leading fields where the results are quite observed, developed, and hence these fields are also aggressively developed with promising results. Over the length of my 30 years service in the field of education, I have seen the revolutionary changes in the field of research with the help of various methods and tools used with more accuracy. The confidence level of new researcher is now have been observed tremendously developed, and their interest in the field of research is also increased. It's a good sign for the development of science and technology. The society is all the time benefited. The concluding remark of the earlier research is nothing but the stepping stone or beginning step for the new researchers for the further research in the same field. I am very much thankful to the organizers who have opened this door and exposed the importance of statistical analysis for the academics and research. Thank you.
Thank you, sir, for your valuable inputs for our FDP, which was really good to know that we have traveled a lot and we have come a long way. On the thought, we will be inviting Dr. Ashok Bankar, Assistant Professor, Department of Microbiology, MES, Abhasab Gharware College, Karve Road, Pune, affiliated to Savitribai Phule, Pune University, for his vote of thanks for the FDP. I welcome you on board, sir, and please take over.
Thank you, Alia,
Doc-
madam. Today is the last day of 1 week FDP on statistical analysis in academics and research. So although it is a last day, but you gain so much feast of knowledge, and you have to digest that knowledge and utilize in your day-to-day life and in your research work as well as in academics. Therefore, I want to give special thanks to all the speakers. They really did very hard work. They also gathered good knowledge, and they put the sequence of good knowledge, and they gave the very good feast of knowledge to all the participants. So therefore, I would like to give the thanks to Mr. Nitin Varse, Dr. Banti Ganguly as a speaker, Dr. Chin Chia, Dr. Sunita Panikar, then Dr. Kishore, Dr. Mohali, Dr. Kishori Kasat, Dr. Shubha Madam, then Suchitra Bhattacharya ji, Mayur Patil sir, Dr. Yogesh Garde, Dr. R.D. Joshi sir, Dr. Firoz Khan, Dr. Yamar Patil, Dr. Parimita Jadhav, Dr. Hitesh Mehta, Hemant Maheshwari, Dr. Ashok Bankar, Dr. Yasar Bamne, Dr. Vijay Padde, Dr. Mahesh Khoth, Dr. Ramakrishna, Dr. Subrata Hati. So all these speakers, they gave very good contribution, and they presented all their data and their knowledge related to statistical analysis and their presentation was really very good, and that's why this 1 week FDP program was successful, and it was very useful for all the participants. Then I want to give the special thank to our anchor. So throughout whole week, um, uh, Dr. Alia madam um, did very well job. So I want to give them very special thanks for the anchoring and for introduction of speakers, as well as for question answer sessions. There are many other faculty members were contributed nicely. Dr. Shital Banerjee madam is there, Pratima Pawar madam, Minal Majeti, as well as the Amol Jadhav sir. All these anchors contributed for this 1 week FDP program. Then I want to give the special thanks to International Journal of Microbial Science, Chief Editor Dr. Sanket Joshi sir, as well as the editor Professor Dakhne sir, and the convener of this program, Dr. Dakhne sir, has organized very nicely. So I would like to highlight the quality of Professor Dakhne sir. Sir really did very hard work for this 1 week FDP program. Sir connected all these people from the different background, from the zoology, from microbiology, from statistics, as well as from biochemistry, from molecular biology. And this is very good personality I found, that is Dr. Dakhne sir, and they connected all these people, and because of his hard work, as well as the positive attitude, as well as the good management, and as well as the he also took the presentation, I mean mock presentation of each and every speaker. So he spent so much hard work, as well as spent so much time, and that is the reason, this is the reason for the success of this program. So for any success, we need very hard work, as well as the good practice, as well as we need to be very positive with good approach. So again, I would like to give all the thanks to our collaborator, that is Sushila, Shankarrao, Gadhave College, Mahavidyalaya from Satara. So in collaboration with this college or Mahavidyalaya, we carried out in collaboration with International Journal of Microbial Science. So again, not least but last, I would like to give thanks to all our participants for their patience, as well as their very good attendance, as well as their nice questions answering sessions. And finally, I give, if I forgot any name, so even I could not took any name in this session, I would like to give thanks to each and everyone, technical session, and everyone, those people who are not in the list, or they are in the background for the support of this FDP program. And I am sure that this program is very useful to each and every faculty member, those who have participated in this program. So I think this is a big success for Professor Dakhne sir, and in future also, so many such kinds of the program Professor Dakhne sir should arrange, manage, and his good management, as well as the Alia Naz madam also did very good job. I have seen that she did very hard work, as well as her presence of mind is very good, and her punctuality, as well as her hard work and her dedication. I give thank you once again to Dakhne sir and Alia Naz madam. Thank you very much.
Thank you, sir, for your kind words, and it will definitely increase my morale, and it will definitely boost and be encouraging to me as well to perform more better. And all good things comes to an end, and when it comes to an end, there is again a new beginning. So before we wind up the session, I have some notifications for the participants. So all the participants very dedicatedly have taken all the 7 days seriously and learned whatever knowledge to acquire from the speakers and the presentations. So now it is the time for the participants to show how much they have gained the knowledge. And for that, we have a MCQ test on November 8, 2020, sharp 7 p.m. The link will be opened or enabled on the official website of www.theijmspublic.com, and the link will be disabled thereafter at 8 p.m. sharp. So keeping that time in mind, please be punctual on logging in. And the links have been able in three different links because all the participants at one go can participate. So this is one thing. And the next thing is three toppers among participants will be awarded with a national award, and the three best speakers, which will be again selected by the peer review and the feedback of the participants, will be getting the best speaker awards also. So hoping to see and meet again and collaborate again for a new kind of learning process. I wish you all the best for your future endeavors. Before I wind up, I would like to tell you all, if you want to have a happy ending, that depends, of course, on where you stop your story. So you can think this is your journey towards your research and towards your learning experiences. With this thought, I would like to wind up this online FDP of ours, which was a great learning experience for me as well personally, because this was something new for me as well. And I am grateful always to the IJMS, that is International Journal of Microbial Science, who, in collaboration um, with Sushila Shankarrao Gadhave Mahavidyalaya, has done a tremendous job to bring in all the aspects of statistics in one roof or in under one umbrella. And this has given us a very good understanding of statistics. And I personally thank you all for being patiently attending all the classes, all the FDPs throughout the session for a week. Thank you all, and I wish you all health, wealth, and prosperity. Stay home, stay safe.